Okay, so the title of my talk will be Aberrant Splicing in B-cell acute lymphoblastic leukemia. And just some background information. Leukemia is the most commonly diagnosed cancer in children. 3 out of 4 are diagnosed will have acute lymphoblastic leukemia. And about 85% of ALL will be of B-cell origin, arising from CD19 positive and surface IgM negative cells. And the hallmark of ALL is genetic and cytogenetic abnormalities, or translocations. So what's interesting is that the prevalence of mutations of tumor suppressors and oncogenes is very low in pediatric ALL. So when you look at the COSMIC database, which is a database of somatic mutations, the vast majority of these driver genes is less than 10%, the actual frequency of mutations. And so we want to ask if there can be a mechanism, a non-genetic mechanism for their deregulation. And previously, as you heard, in other leukemias, there are mutations that occur in splice factors, but in ALL, there are no such mutations that we see. So we first were introduced to the splicing world by discovering an alternative isoform of CD19. And what's interesting about this isoform is that it skips exon 2, which contains the epitope for CART19 immunotherapy. And we also discovered that SRSF3, this splice factor, promotes exon 2 inclusion. So we wanted to ask, are there global levels of splicing deregulation? And so for this, we needed appropriate controls. So we isolated and sorted human B-cell fractions into early B-cell or early progenitors, pro-B, pre-B, and immature B-cells. And this is a PCA plot of RNA-seq, and we see that they're nicely cluster with their B-cell fractions. And so the question is, where does ALL cluster? And we had 18 in-house leukemias, and we saw that they clustered very nicely with the pro-B, with the pro-Bs. And when you look at CD19 and CD34, which are pro-B markers, we see that the ALL samples had those. So the appropriate comparison that we did with splicing in the context of splicing was pro-B versus ALL. So with the help of the Barash lab and in close collaboration with them, we ran the Majik algorithm. So how this works is that you have 2 conditions. In this case, we have the normal pro-Bs versus our leukemias. And then it defines LSVs. LSVs stand for local splice variation. And then we can quantify them and then call it differential LSVs. So this is an example. In this case, we see this LSV with 3 different splicing events. We see that this exon can be skipped, or both these exons can be skipped, and so forth. So for this paper, for this study, we focused on exonic events and any change that was at least 20%. And so when we looked at global levels of alternative splicing, we saw that, so what we did was we had 18 samples and we made 18 pairwise comparisons to the pro-Bs. So what we saw is that the vast majority of LSVs, or differential LSVs, were unique to a specimen, kind of illustrating the heterogeneity of ALL. But what we also saw that a total of 279 aberrant LSVs and 241 unique genes were detected in all 18 pairwise comparisons. And so when we did a GO set enrichment analysis, a gene set enrichment analysis, we found that it was enriched for RNA splicing. So the next question we asked is, were splice factors themselves misspliced? And what we saw is that we found 101 splice factors that were misspliced in ALL. And then when we compared this back to the different states of B cells, we only saw anywhere from 6 to 19. And this suggests that this missplicing of splice factors themselves may be responsible for this widespread deregulation of splicing in ALL. So this is a very, very simple schematic, but the take-home message of this is that there are SR proteins that primarily act as enhancers, so they promote exon inclusion. And then we also see HNRMPs, which usually act as silencers. There's many exceptions, but this is a general overview. And so the question is, do we see any missplicing of SR, the HNRPs, in our samples? And so here's a heat map. So this is the SRSF family, and this is the HNRP family. And what we see is here, so this is delta psi between the probees, the normal probees, and ALL. And these are the 2 normal samples and the 18 leukemias. And what we found was that there was multiple aberrant splicing events happening in multiple genes. And in some cases, for example, SRSF7, there are 2 different splicing events happening. But what's interesting is that HNRPA1 was the most prevalent, so we decided to focus on this particular splice factor. When you look closely at it, we see that this is the actual LSV, and this is a violin plot of the percent spliced in. So what we see is that exon 11 is usually included in the probees. It's almost included 90% of the time. However, in ALL, this is one representative sample. It goes down to 40%. So essentially, this exon is skipped to exon 12 or 13. And when we map this back to the genome, we see that this actually corresponds to the 3' UTR. And because it corresponds to the 3' UTR, we think this may be affecting the stability of the RNA transcripts. And so when we did correlation between expression and exon 11, using RT-qPCR and RNA-seq data, we see a positive correlation. And so with this positive correlation and this stop codon presence, we believe that the skipping of exon 11 may decrease RNA steady state levels, and we think it's through NMD or nonsense mediated decay. And so what we did then was we knocked down UPF1 and UPF2, which are known NMD factors. And what we saw is that when we measured the transcript level expression, we saw that the skipping to exon 12, that transcript accumulated when we knocked down these NMD factors. So the skipping event that we primarily observe in ALL, we believe decreased RNA steady state levels of HNRPA1. And so what we did then was we knocked down this splice factor, and here are just validations. This is expression, and this is a Western blot showing the protein levels are knocked down. So then what we did then, we essentially found 184 genes that were misspliced between the control and the knockdowns. So we called these targets. And we were able to find 213 different or differential LSVs. So then when we overlapped it with our events in ALL, we found that 1/3 of these LSVs were also LSVs that were misspliced in ALL. And so we were able to define HNRPA1 dependent LSVs. So then the next question is, what are these genes that are misspliced? And what popped up to us were tumor suppressors Dicer1 and NT5C2. So in Dicer1, we see that in normal cells, the blue event, which is the skipping of exon 4, is usually more prevalent, while in ALL, it goes down to 40%. And when you map this event back to the genome, we see that it corresponds to the 5' UTR, which potentially diminishes its translation efficiency. When you look at NT5C2, we see that exon 11, which is usually skipped, is suddenly included in ALL. And it's interesting because this is actually an in-frame coding exon. And so then we looked at other tumor suppressors just in general, which ones were misspliced. We found about 40 or so with 80 or so LSVs. Here's Dicer1. We also found FBXW7, P53, and so forth. And then to confirm this in larger data sets, we looked at Target and St. Jude's, and we found 81 LSVs, and we were able to recover all of them except for 2 in Target and St. Jude's. And here's an example of P53, which is also experimentally validated. So in P53, in normal cells, we see that this exon 9I is skipped, while in ALL it's included. So it goes from 11% to 60%. And in solid cancers, this alternative isoform has therapeutic relevance because it essentially makes it resistant to chemotherapy. So looping back around, when we compare the frequency of mutations against the frequency of LSVs, we see that in the majority of cases, the LSV frequency is much, much higher than somatic mutations, suggesting that these driver genes may be preferentially affected by post-transcriptional mechanisms such as alternative splicing. And so in conclusion, we see there's systematic deregulation of splicing in ALL. One of its causes may be the missplicing of HNRPA1 and other splice factors. And we also show that there's abnormal splicing of tumor suppressors in oncogenes that may contribute to the disease. And here's the acknowledgement slide. Katie Black did the majority of the wet lab. Katerina Heyer also helped with the bioinformatics. My co-mentor, Deanna Taylor, and then our collaborators, Kristen Litch and Joseph Barrage, and the pathology for providing us samples. Thank you. Yes. So here the question was, were we able to find healthy individuals for control? So yeah, so this analysis, we compared it to normal healthy donors that we found at CHOP. We had 4, but we're getting more as we speak.
Any other question?
So where you got to begs the question, what's causing those changes, right? Like these ones.
So what's causing those changes in splice factors? The short answer is we don't know, but we suspect that there may be upstream kinases that may be mutated that may be affecting these splice factors. We're investigating that as we speak.
So the symmetrical question is, what are they doing downstream? Right? So you show P53 changes. So what is that doing P53 function, for example?
So in that case, so we're not sure what changes, what's the functional relevance of these changes downstream. So we're not sure about P53, but for example, NT5C2, it's a nucleotidase that's responsible for the cleavage of the 5' end of nucleotides. And we're essentially what we're doing is we're using morpholinos to block exon inclusion to see if there's functional relevance to that. So that's one case.
Okay. Thank you very much.